Okay, imagine this.
Yeah.
You step into the future,
Right.
and your clothes, they're not just, looking good. They're actively doing something for you.
Hmm.
Picture medical textiles, maybe your running gear, that naturally fight off bacteria. Not with, harsh chemicals,
Yeah.
but with these amazing, vibrant colors made by tiny microbes.
It sounds like science fiction, but it's closer than you might think.
Exactly. That's the fascinating possibility we're diving into today. We're taking a deep dive into the world of bacterial pigments, these natural colors bacteria make, and exploring their, incredible potential.
Right, as sustainable functional agents for textiles, and especially their antibacterial side.
Yeah, those antibacterial superpowers. And our guide for this deep dive, it's actually this really comprehensive academic review paper. It pulls together a ton of research on the challenges, but also the really exciting future here.
It's a great overview of the field.
So over the next little while, we're going to unpack the science, right? How bacteria actually create these pigments. We'll look at the clever ways researchers are getting them out.
The extraction methods. Yeah.
And how they're being put onto fabrics, and really understand why they work so well against bacteria.
And we shouldn't forget the hurdles either.
Definitely not.
Yeah.
We'll tackle the big hurdles and, peek at what the future might hold. Okay, let's get into it.
It really is amazing stuff. I mean, when you think bacteria, you often think, germs, illness.
Right. Negative stuff.
But they are nature's tiny chemists. They create this huge range of compounds, and many have properties way beyond just color. These pigments, they're often part of the bacteria's own survival kit.
Oh, interesting. So they're not just decorative for the bacteria. They have a purpose.
Exactly. Maybe protection, maybe communication, maybe interacting with their environment and how they make them.
Well, it's down to these incredibly complex little factories inside their cells.
Biosynthetic pathways, the paper calls them.
Right. Intricate, multi-step processes. And what colors they make, or how much, can really depend on their surroundings. Things like temperature, pH, what nutrients are around. Okay, and the paper we're looking at, it focuses on 4 key pigments that seem especially promising for textiles. Can you maybe walk us through those?
Sure. We'll look at piocyanin, carotenoids, melanin, and prodigiosin. They're all chemically quite different, come from different bacteria, and importantly, they tackle microbes in different ways.
Okay, let's start with piocyanin. That's the blue-green one from Pseudomonas aeruginosa, right?
Yeah.
The paper gets pretty technical on the pathway, but what's the key thing to know about how it's made and why it's interesting?
Well, the main thing really is that Pseudomonas makes it through a whole series of enzyme-driven reactions. Think of it like a mini assembly line.
Yep.
But what's really fascinating is that this pigment, it's a redox molecule. It can easily gain or lose electrons.
Oh, like it juggles electrons.
Exactly. And that ability allows it to mess with cellular processes, both in other bacteria and even in, say, our own immune cells. It does this by generating these damaging molecules called reactive oxygen species, ROS.
ROS, that's familiar. Like oxidative stress, cellular rust? Is that the idea?
That's a great analogy, yeah. It causes oxidative stress. And in terms of fighting other bacteria, the paper shows it's particularly good against gram-positive bacteria.
Got it. So piocyanin, blue-green, electron juggler, makes ROS good against gram-positives. Okay, what about carotenoids? We see those colors everywhere.
Absolutely. Carotenoids give us those fantastic reds, oranges, and yellows found in lots of bacteria. Flavobacterium, Micrococcus, Serratia.
And how do they make those? Is it a similar assembly line?
It's based on building blocks, these isoprenoid units. They kind of string them together using specific enzymes to create these larger, colorful molecules.
Like molecular Legos.
Precisely. And interestingly, like other pigments, bacteria often ramp up production when they're stressed. High temperature, saltiness, or notably high light intensity can trigger it. Sometimes it's a form of sunscreen for them.
Makes sense. And the paper suggests these target gram-negative bacteria like E. coli. How do they work differently from piocyanin?
They tend to interact with the outer layer of gram-negative bacteria, specifically with proteins called porins.
Porins. Are those like the gatekeepers in the cell wall?
Exactly. They control what gets in and out. Carotenoids seem to mess with these porins, disrupting that outer barrier, making the whole membrane more leaky. Stuff leaks out that the bacteria needs.
Okay, messing with the gates. Got it. Next up was melanin?
Yep, melanin. Usually that familiar brown or black color, though bacterial melanin can have other shades too. It's derived from simpler starting materials, amino acids like tyrosine.
And the bacteria just link those tyrosine units together somehow.
Essentially, yes. There's an oxidation step, and then the resulting molecules kind of link up spontaneously. It's called autopolymerization to form these large, dark polymers. You find it in quite a few different bacteria.
And can they make more if needed?
Yeah, researchers found they can boost production just by adding more tyrosine or even protein-rich stuff to the grape medium, feed them more building blocks, basically.
Clever. And how does bacterial melanin fight other bacteria? Does it also use that ROS trick?
It does, yeah. Its chemical structure allows it to generate ROS, just like piocyanin. But it has other weapons, too.
Oh.
It can directly interact with the bacterial cell membrane, disrupting it. And this is really cool. It can have a photothermal effect.
Photothermal, light and heat.
Exactly. It absorbs light energy and converts it into heat. Enough heat right there can be lethal to nearby bacteria. And it works against both gram-positive and gram-negative types.
Wow, heating them up. That's pretty neat. Okay, finally, protegiocin. That's the really intense red one.
That's the one. A very distinctive red pigment. You often find it in bacteria like Serratia marcescens. Its production pathway is a bit unusual.
How so?
The bacteria actually build two separate precursor molecules, and then a specific enzyme acts like a molecular stapler, joining them together to make the final red protegiocin molecule.
A two-part construction. And this was highlighted for strong antibacterial action, mainly against gram-positives again.
Yes, primarily gram-positives. Protegiocin is quite hydrophobic. It doesn't like water, prefers fatty environments.
Like a cell membrane.
Precisely. So it integrates itself into the bacterial membrane. But instead of just poking discrete holes, it causes a more general disruption, making the whole structure unstable and leaky. The paper calls it chaotropic disruption.
Chaotropic.
Yeah.
So like it makes the whole membrane structure just fall apart rather than punching holes.
That's a good way to picture it, yeah. Less like a punch, more like making the wall just crumbly. Essential things like ions, proteins, sugars leak out. Plus, it messes with other vital stuff like respiration and cell division.
The crumbly wall analogy helps. Okay, so 4 pigments, 4 different ways bacteria make them, and 4 distinct strategies for fighting other microbes. ROS damage, messing with gates, heating things up, making walls crumble. That's quite a diverse chemical arsenal.
Nature's toolkit is pretty impressive, and understanding these mechanisms is crucial, but it's only the first step. The next big question is, how do we actually get these molecules out of the bacteria to use them?
Right, extraction. The paper lays out the basic steps. Break open the cells, get the pigment out, purify it, check what you got. And getting that first step right, breaking the cells efficiently, seems key for getting a good yield.
Definitely. Broadly, you've got mechanical methods and chemical methods. Mechanical ones physically rake the cells. Think bead milling, basically grinding them with tiny beads. Or high-pressure homogenization.
That sounds intense, like putting them through a microscopic pressure washer.
Sort of, forcing them through a tiny valve at incredibly high pressure. These can be good for large scale, but they tend to use a lot of energy. And, you end up with a lot of cell debris you need to clean up later.
Any other mechanical ways?
There's ultrasonication, using high-frequency sound waves to create tiny bubbles that collapse violently, breaking the cells. It's lower cost, potentially, but you have to be careful not to degrade the pigment itself with too much energy.
Okay. And then the chemical methods, often using solvents.
Right. Using liquids to dissolve the pigment out. The best solvent depends on the pigment. Some are more water-soluble, some are oil-soluble. You often warm it up a bit to help, but again, too much heat can destroy the pigment. Solvents are common because they can be, well, simple and relatively cheap.
But the paper also mentioned a big push towards green chemical methods, which sounds important for sustainability.
Hugely important. This includes techniques like supercritical fluid extraction, or SFE.
Supercritical. What does that mean?
You take something like carbon dioxide, CO2, and put it under high pressure and temperature. It enters this weird state where it's kind of like a liquid and a gas. This makes it really good at dissolving things and getting into tight spaces, like inside cells.
So you use pressurized CO2 to pull the color out?
Pretty much. It's fast, efficient, uses a non-toxic solvent, and you can just release the pressure to get your pigment back and reuse the CO2.
That sounds very neat. What else is green?
There's microwave-assisted extraction, MAE. Uses microwaves to rapidly heat the solvent that's inside the cells, making extraction super fast and using very little solvent overall. The equipment can be expensive, though.
Yeah.
And enzyme-assisted extraction, EAE. Using specific enzymes, like molecular scissors, to gently break down the bacterial cell walls. This is great for delicate pigments because it uses mild conditions. Plus, sometimes you can get these enzymes from waste products and even recycle them.
Using waste to get color out of microbes possibly grown on waste. That's like circular economy squared.
It has that potential. And finally, ionic liquid-assisted extraction. Using these special salts that are liquid at room temperature or slightly above.
Liquid salts.
Yeah, they have unique properties, low flammability, and you can sometimes design them to be really selective for the pigment you want, potentially giving higher purity. They can be reused, but the initial cost is high, and handling them needs care.
Wow, okay. So a whole range of ways to get the pigments out, from brute force to quite sophisticated, greener methods. Researchers are clearly thinking hard about efficiency and environmental impact.
Absolutely. Because once you have the purified pigment, then comes the really exciting part, putting it onto fabric.
Dyeing with bacteria.
Exactly. And bacterial pigments offer this incredible rainbow of colors. They can bind pretty well to textile fibers too, often through ionic interactions.
Like tiny chemical magnets, positive on the fiber, attracting negative on the pigment, or vice versa.
That's often the principle, yes. But how well it works, how much color the fabric picks up, how strongly it holds depends on a lot of factors. The type of fiber, is it cotton, wool, nylon, the pH of the dye bath, the temperature, how long you leave it in.
Right. And the paper gives loads of examples for our key pigments. Let's revisit carotenoids, the reds, oranges, yellows. Where have people tried using those?
They've been put on all sorts of things. Natural fibers like silk, linen, cotton, but also synthetics like rayon. Researchers are finding bacteria that make them everywhere. Soil, seawater, even industrial wastewater.
Wow.
For example, a yellow pigment from a soil bacterium called Chrysobacterium worked well on silk and cotton, dyeing them at about 80-90 degrees Celsius. Another red one gave a deep orange on cotton at 70 degrees.
And you mentioned 50 to 90 degrees. That sounds relatively low temperature for dyeing, doesn't it? Which would be good for energy saving.
It often is, yeah. Lower temperatures generally mean a better environmental profile. Then there's melanin, the brown-black one.
Putting bacterial melanin onto textiles, that feels unusual.
It's less common, but definitely being explored. Studies have dyed wool and cotton. One pigment from a Streptomyces species dyed onto wool actually showed pretty good fastness properties.
Fastness meaning how well the color lasts.
Exactly. How well it resists washing, fading from light, rubbing off, or reacting to sweat. The study reported levels 4 to 6 on standard scales, which is approaching commercially acceptable levels for some aspects.
That's promising.
And treating cotton dyed with melanin using an enzyme called lactase significantly improved how well the color stuck after washing. So there's potential there for unique colors.
Okay, what about violaceum, the bluey purple one?
Violaceum, yeah, often from Chromobacterium violaceum. Early work tried it on natural and synthetic fibers. Nylon seemed to take the dye quite well, but the fastness was variable, especially light fastness.
Ah, the fading problem again.
Yes, that seems to be a recurring theme. Light fastness often scored quite low, like level 1 or 2 on a 5-point scale, meaning it would fade very quickly in sunlight. Not ideal for many applications.
So a beautiful color, but maybe not for your summer wardrobe.
That's been the challenge, but researchers are getting creative. There's work on simultaneous fermentation and dyeing, basically, growing the bacteria with the fabric in the broth.
Dyeing it as it's made.
Kind of. On synthetic fabrics like polyamide, this gave really vibrant purples and actually achieved excellent wash and perspiration fastness, level 5, the top score.
Level 5 wash fastness is fantastic, but the light fastness, any improvement there?
Unfortunately, still poor in many reported cases, often levels 1 to 3. Even compared to standard synthetic dyes, violacine really struggled with light stability in some studies. People are trying things like adding nanoparticles, but it remains a major hurdle for wider use.
Okay. And prodigiosum, the red one. The paper seemed to mention that one quite a bit in the context of textiles.
It's definitely one of the most studied bacterial pigments for dyeing, probably because it's relatively easy to produce from bacteria like Serratia, and it gives that strong red color.
Were there stability issues there too?
Early on, yes, especially with high temperatures during dyeing. But later studies found ways around that. They achieved good color yield and decent fastness on cotton and wool, particularly when using mordants.
Mordants. Those are chemicals that help the dye stick better, right? Like a primer for the fabric.
Exactly. They help form a stronger bond between the pigment and the fiber. Prodigiosum has dyed lots of different fibers. It holds up okay in acidic or neutral conditions, sometimes even against bleach or heat, but yes, light resistance is often its Achilles heel.
Seems to be a pattern.
It does. Though some studies optimizing the dyeing conditions, especially on natural fibers like silk, reported much better fastness results across the board, including light fastness scores of 4-5.
Oh, so maybe the fiber type makes a big difference for prodigiosin. Silk performs better than cotton.
In those specific studies, it seemed silk had a higher affinity and showed better overall fastness. There's also been really interesting work using surfactants, soap-like molecules during fermentation to help extract the pigment, then applying it as a nanosuspension.
Nanosuspension. Tiny particles of pigment.
Exactly. Applying this to acrylic fabric, even at high temperature, gave a deep color and demonstrated antibacterial function right on the dyed fabric.
Color and function in one go. That's really the goal, isn't it?
It absolutely is. And studies comparing prodigiosin on cotton versus synthetics like chiffon found the synthetics often showed better wash fastness. Excellent was the term used, but the light fastness was all over the place. Very good on some, poor on others. It really highlights that light stability is a major, complex challenge for many of these pigments.
Right. So we're seeing amazing colors, successful application to fabrics, but that durability, especially against sunlight, is a consistent issue needing more work.
Right.
But like you just mentioned with the nanosuspension, the goal isn't just color.
Precisely. Beyond the aesthetics, a huge driver for this research is giving textiles antimicrobial properties. We already talked about how pigments like prodigiosin and phycyanin can disrupt bacterial membranes.
Yeah, their natural ability to interact with those fatty cell walls makes them prime candidates for fighting germs.
Exactly. And the paper gives some pretty compelling examples. That prodigiosin on acrylic fabric. It showed an 81.8% reduction against Staphylococcus aureus, a really common and often nasty bacterium.
That's significant. And on other fabrics.
Yes, the nanosuspension on the lyocell showed over 99.9% reduction against S. aureus. Even just a simple prodigiosin coating on cotton showed strong activity. You could see a clear zone around the fabric where bacteria just couldn't grow.
Like a little protective halo.
Kind of, yeah. And red prodigiosin from marine bacteria applied to cotton in synthetics showed this antibacterial effect lasted. Synthetics like chiffon, despite maybe having light fastness issues, showed really high germ reduction, over 95% against both E. coli and S. aureus. Linen also did well.
Wow, over 95%. That's serious germ-killing power.
It really is. And it's not just prodigiosin. Pigments from soil bacteria, including carotenoids and pyocyanin, showed activity, with green pyocyanin extracts being particularly potent against both gram-positive and gram-negative bacteria in lab tests.
The paper even mentioned a blue-green pigment from P. aeruginosa on silk, showing strong inhibition against S. aureus and other pathogens. And interestingly, it pointed out that just having more dye on the fabric didn't always mean better antibacterial action.
That's a really important nuance. It's not just about staining the fabric. The pigment needs to be applied in a way that keeps it functional. Streptomyces bacteria are another major source people are studying for antimicrobial pigments.
Right.
One from Streptomyces cyanea on silk showed 95% activity against S. aureus. A yellow one on cotton polyester blends also showed promise. Studies testing crude extracts from multiple Streptomyces strains found activity against skin bacteria and yeast, too.
So the evidence seems really strong across different pigments, different bacteria, different fabrics. These colors can genuinely turn textiles into germ fighters.
Which naturally brings us to the really critical questions.
Yeah.
Are they safe? And how do they stack up environmentally? Especially now with everyone looking for greener, safer materials.
Yeah, biocompatibility has got to be paramount. Safe for us, safe for the planet.
Absolutely. The good news is, being natural products, bacterial pigments are generally biodegradable, but testing is vital. The paper mentions a study on a bacterial melanin pigment which found it wasn't toxic to human skin fibroblast cells, even at fairly high concentrations. That's a positive sign for things that touch our skin.
Okay, that's good. And for the bigger environmental picture, you need that life cycle assessment, the LCA, right?
Exactly. LCA is the tool for systematically looking at the entire environmental footprint, from cradle to grave. Growing the bacteria, extracting the pigment, dyeing the fabric, using the textile, disposing of it. All of it.
And where do the big environmental impacts tend to show up in the LCAs done so far?
Often it's the cultivation stage growing the bacteria. That can be energy intensive. Think fermenters, sterilization, drying processes.
Right.
They all use electricity.
Right.
But the paper makes it clear that there's huge potential for improvement. Optimizing how you grow them at large scale, using more efficient drying, maybe growing them in open ponds instead of closed reactor where possible.
And switching to renewable energy sources.
That's a massive factor. Switching to renewables for that electricity demand can dramatically slash the footprint. Some estimates suggest you could cut the impact by like 85% just by focusing on improving cleaning, sterilization, and drying steps and using clean energy.
85%. That's huge. How do they compare to traditional synthetic dyes in these LCAs?
It's still an emerging area, and comparisons are tricky. One study comparing a microalgal pigment to a synthetic one found the natural one initially had a higher impact, mostly due to electricity for growing the algae. But using renewable energy brought that impact way down.
Okay.
Another study looked at astaxanthin, that reddish pigment, comparing production via bacteria, algae, and chemical synthesis. It found that chemical synthesis could have a lower impact overall. BUT bacterial production, especially if powered by renewables, could actually match or even outperform the albal route in key environmental categories. It highlighted that the fermentation step itself is the key place to optimize.
So the takeaway is, microbial production has an energy cost, but it's one that can be significantly reduced, potentially making them environmentally competitive with, or even better than, other options, especially with clean energy.
That seems to be the conclusion, yes. LCAs are vital tools to guide the industry towards the most sustainable pathways, and to make the case for these pigments as genuinely eco-friendly alternatives. But, it's not all perfectly solved yet.
No, I imagine there are still some pretty big challenges before we see bacterial dyes taking over the textile world. What are the main hurdles?
Well, number one is probably scaling up production. Meeting the massive global demand for textiles means needing way more pigment than labs currently produce, and doing it cost-effectively.
Ah, the cost factor. Is it much more expensive than synthetic dyes right now?
It often is, yeah. The paper mentions microbial beta-carotene costing maybe double what synthetic beta-carotene costs per kilogram. Part of that is the cost of the growth media, the food for the bacteria if you're using expensive synthetic stuff. And the extraction process can add significant costs, too.
Okay, so bringing the price down is critical. What else?
Regulatory compliance and just acceptance. Getting approval to use these, especially if you're making antimicrobial claims, involves navigating really strict regulations in different parts of the world. You need a lot of safety data.
And you mentioned acceptance.
Yeah, consumer acceptance. Are people comfortable wearing clothes dyed with stuff made by microbes? And industry acceptance too. Manufacturers need convincing.
That's a good point. The potential ick factor, maybe? Plus industry inertia.
Could be. Then there's stability. We talked about light fastness, but bacterial pigments can also be sensitive to heat, pH changes. They're natural molecules, often less robust than synthetics. This makes standardization difficult too, ensuring every batch has the exact same color and performance when biology is inherently variable.
Right. Consistency is key for industry.
Definitely. And while nature offers diversity, the range of commercially viable, stable, and scalable colors from bacteria is still more limited than the vast palette of synthetic dyes currently available. Plus, optimizing those extraction and purification methods to be cheaper, greener, and more efficient is an ongoing challenge.
And just getting the bacteria themselves to be super producers, reliably pumping out high yields.
Exactly. Strain improvement and controlling the fermentation conditions perfectly are crucial for successful scale-up. And finally, just plain old industry awareness. Many in the textile industry just don't know much about these alternatives or perceive synthetics as easier and cheaper to work with.
Okay, so quite a list. Scale, cost, regulation, acceptance, stability, standardization, color range extraction, optimization, awareness. But the paper must offer some potential solutions, right? What's being done?
Oh, absolutely. Researchers are attacking these problems from all angles, improving production yields and even creating new colors using genetic engineering. Tweaking the bacteria's DNA is a big focus.
Reprogramming the bacteria.
In a sense, yes, or finding clever ways to boost yield with supplements. The paper mentions one study where adding simple peanut powder to the growth medium increased prodigiosin yields 6-fold.
Peanut powder. Seriously.
Sometimes simple works. Optimizing the whole fermentation process temperature, pH, feeding strategies is key, and improving recovery, using things like that ultrasonic treatment mentioned earlier to get more pigment out efficiently.
Okay, and for the regulatory and acceptance side?
More research, essentially. Building that robust database of safety and efficacy data that regulators need, and fostering collaboration, getting researchers, industry players, and regulatory bodies talking to each other more effectively.
Makes sense.
Raising industry awareness is also vital, showcasing the benefits, maybe using those LCA studies to demonstrate the environmental advantages over conventional methods, and of course continuing to develop and refine those greener extraction techniques like EAE and SFE.
It really sounds like a dynamic field, tackling challenges from the microscopic level right up to market adoption.
It absolutely is. And looking ahead, the potential here is just immense. Bacterial pigments could genuinely reshape industries, especially textiles. I mean, bacteria are practically an inexhaustible resource for novel bio-based materials.
The future opportunities seem huge, driven by better technology, consumers wanting sustainable options, and that need for materials that do more.
Definitely. And think about the specific applications in textiles, combining color and that antibacterial function we talked about. The paper highlights several areas. Medical textiles is a huge one.
Hospital gowns, bedding, wound dressings that actively fight infection.
Imagine the impact on healthcare-associated infections. Or sportswear, naturally antimicrobial, reducing odor without chemical treatments. Sustainable and functional.
And by that, what else?
Home textiles. Towels, bedding, maybe even carpets with enhanced hygiene, resisting mold and mildew, potentially lasting longer.
Could they even end up in food packaging, like you hinted?
That's another potential avenue, inhibiting spoilage microbes, extending shelf life naturally. Outdoor gear, too, preventing microbial growth on tents, awnings, outdoor furniture, making them more durable.
And just fashion, offering unique, sustainable colors with that built-in antimicrobial benefit.
Exactly. So really the big picture here is that bacterial pigments aren't just natural dyes. They are truly multifunctional bio-based solutions. They offer a pathway to creating materials, especially textiles, that are more sustainable, safer, and higher performing across an incredible range of uses. It's about using nature's chemistry to weave function directly into the fabric of our lives.
Wow. That really takes us from the tiny world inside a single bacterium all the way to the clothes we wear and the materials all around us. We've covered how bacteria make these colors, how scientists get them out, how they're put onto fabrics, and critically, how they can fight off germs.
It's quite a journey. This deep dive really underscores how harnessing the power of microbial life can provide maybe not a shortcut, but a smarter path towards innovative, sustainable, and functional textiles. For everything from saving lives in hospitals to making our gym clothes smell better.
It definitely changes how you might look at microbes, and maybe even your t-shirt. So here's a final thought to mull over. As you think about the future of materials, could these vibrant microbe-made colors, with their built-in defenses that we've discussed, be a key part of creating a cleaner, greener, maybe even healthier world, quite literally woven into the fabric of our daily lives?